Investigation number 4, diffusion and osmosis. We're going to start with procedure 1, surface area and cell size. Begin by placing some phenolphthalein into 2 test tubes. Next, we're going to add 0.1 mole HCl to 1 test tube. Then we'll swirl to mix the solutions. Using the same procedure, we're going to add 0.1 mole NaOH to the other test tube. Swirl and observe. Investigation number 4, modeling diffusion and osmosis. We're going to start with our dialysis tubing, and we are going to place 10 milliliters of sodium chloride inside. We're going to make sure that you leave some room for water to be able to absorb in. So this is our first dialysis tubing filled with sodium chloride, and we're going to weigh. So our initial weight is 12.227 grams. We are now going to place our dialysis tubing into our container of water, and we're going to let it sit for 30 minutes. We're going to fill our second dialysis tubing up with glucose, 1 mole glucose solution. All right, and we are going to place this on our scale and record for our initial weight. And our initial weight for our glucose is 14.220 grams. And we're going to place our glucose dialysis tubing into our container of water to sit for 30 minutes. Our third dialysis tubing, we are going to put 1 mole of sucrose solution in. You now weigh our glucose, or sucrose, we're on sucrose, yep. And our initial weight for our sucrose is 15.583 grams. And we're going to place our sucrose into our container of water to sit for 30 minutes. Fourth dialysis tubing is albumin powder. We're going to weigh our albumin powder in our dialysis tubing. And our initial weight is 14.258 grams. And we are going to place it into our container of water to sit for 30 minutes. Fifth dialysis tubing is going to be our control. And we are just putting 10 milliliters of water in there. Our water. And our initial weight is 11.959 grams. And we are going to place it also in water to sit for 30 minutes. Okay, and so now we're going to take our final weight measurements. This is our NaCl. We're going to place it on our scale. And our final weight is 13.904 grams. We're going to take our next one, which is our sucrose solution. Place it on the scale. And our final weight is 22.612 grams. And we're going to move on to our glucose solution. that on the scale. And our final weight is 18.082 grams. Our third solution is the albumin powder. Our final weight is 15.324 grams. And our final one is our control, which is our water. And our final weight is 11.753 grams. We have our potato cores and our different solutions of sucrose, and we're going to start with our 3 potatoes, and we're going to weigh those. And we have our initial weight at 5.035 grams, and we're going to place those in our solution to leave overnight. We're going to start with our second set of 3 potatoes. Weigh those, and we have 5.030 grams. Place this in our solution of red overnight. We're going to take our next 3. And our initial weight is 4.897 grams. Place these in our blue solution overnight. Next 3. Initial weight is 4.871 grams. Place this in our yellow solution overnight. Initial weight is 5.251 grams. Place this in our green solution overnight. Initial weight is 4.991 grams, placed in our purple solution overnight. The ultimate goal is to figure out the molarity of the sucrose solutions based on the rate of diffusion. Here's day 2 of our potato cores. We're going to start with our first clear solution. We're going to take the 3 potato cores out. Blot, and then we are going to weigh. Our final weight is 3.786 grams. All right, so this is our red solution. Our final weight is 3.196 grams. Our blue solution. Is 3.555 grams. Yellow solution. .312 grams. Green solution. 5.443 grams. And our purple solution. .417 grams. Okay. And as you can see, these tend to be a little bit more shriveled and squishier. The yellow and the green and the purple are much more rigid, not so squishy, more hard. And now we will complete our percent change.